Welcome back to the Deep Dive. Today we're setting out on a specific mission. We want to find the absolute origin story, that single scientific spark behind a medical technique that honestly still sounds a bit like science fiction, hyperbaric oxygen therapy or HBOT.
It does, doesn't it? And HBOT is, well, it's pretty widely accepted now, though still specialized. You hear about it for chronic wounds, radiation injuries, some nasty bone infections.
Right, and emergencies like CO poisoning too.
Exactly, carbon monoxide poisoning, the bends in divers. But when you look at that long list of uses, you kind of have to wonder, where did the basic idea come from? What was the fundamental discovery that showed putting someone under intense pressure with pure oxygen could actually work physiologically?
That's exactly the puzzle we're tackling for you today. Our deep dive is zeroing in on the foundational research papers. We're really determined to isolate that one single seminal paper, the one that gave the undeniable proof of principle for using pressurized oxygen medically.
Yeah, the physiological proof, not just, hey, we tried this and it seemed to help.
Exactly. This is the ultimate shortcut for you listening in to grasp the scientific bedrock, the core reason why anyone ever thought, let's put someone under 3 times normal atmospheric pressure and flood them with oxygen. It sounds extreme.
It is extreme, and that distinction is really crucial. Scientific history isn't always a straight line from A to B. We're not necessarily looking for the paper describing the first time it was ever used on a person or, the latest big clinical trial.
No, we're hunting for the experiment that fundamentally shifted how we understood mammalian survival, how oxygen gets around the body. That's the scientific genesis we're after.
The moment someone proved it was even possible.
It's a really interesting search because often the most important paper, the one that truly broke the conceptual ground, it isn't the one you'll see cited constantly in journals today. It's become a piece of history, almost taken for granted.
Right, its impact is baked into everything that came after.
So if we're digging down the bedrock, we need to start with the architect. And in the world of hyperbaric medicine, there's really one name that stands above the rest. He's widely and correctly known as the father of modern hyperbaric medicine.
You're talking about the Dutch surgeon, Dr. Boerema.
That's the one, Dr. Boerema. His work in Amsterdam, back in the late 1950s, was happening against a backdrop where, well, knowledge was pretty limited, wasn't it?
Oh, definitely. Hyperbaric chambers weren't totally new. You had these things back in the 19th century, often used for, let's say, dubious health claims.
Right, miracle cures and whatnot.
Yeah, exactly. And then more credibly for treating decompression sickness in diverse bends. But those uses were either procedural, like for diving safety, or reactive, treating a problem after it happened. They weren't based on some deep, proactive physiological idea for general medicine.
That's a really important point. Before Boerema, the medical world didn't really think high-pressure oxygen could, say, revolutionize wound healing or help bypass major systemic problems. It was seen as a tool for divers, mostly. So what was Boerema thinking? What put him on this, well, unique path?
Well, Boerema was a brilliant surgeon, an experimental surgeon, and he was thinking about complex heart surgery. This was still early days for that field.
Good.
He realized if he could somehow drastically lower a patient's oxygen needs, maybe through hypothermia, making them really cold, he might buy himself more time to operate. But crucially, he still needed a way to absolutely guarantee oxygen delivery, even if blood flow was minimal or compromised during the surgery.
So it wasn't initially about infections or wounds at all. It was a surgical problem, a logistical problem almost, needing a really creative physiological workaround.
Precisely. And this whole line of thought, this intellectual journey, it really culminated in the definitive work published in 1960. Now, there was an initial publication in 1959 that sort of touched on the concept. Yeah, but the 1960 paper, that's the one we consider the true scientific foundation. It's the one that really solidified the proof.
And it has a title that, even reading it today, it's provocative. It's almost shocking. The seminal publication, Boerema, Manet, Brummelkamp, and colleagues, 1960. The title, Life Without Blood, a study of the influence of high atmospheric pressure and hypothermia on dilution of the blood.
Life without blood. Just think about that. It's an immediate, incredibly aggressive challenge to, well, centuries of accepted medical wisdom.
Totally.
The absolute core belief in medicine was, and largely still is, that red blood cells and the hemoglobin inside them are the non-negotiable requirement for carrying life-sustaining oxygen. To even suggest life could go on without them, that was basically physiological heresy back then.
It was like throwing down an intellectual gauntlet, wasn't it? Saying everything you thought you knew might be wrong. Okay, so let's unpack this. Why that title? What exactly were they trying to prove with such a, frankly, high-stakes experiment?
They chose that title because it was literally what they set out to achieve, or at least demonstrate was possible. The question was stark. If you completely remove the primary biological mechanism for oxygen transport, hemoglobin, can you replace its function entirely using a purely physical force, namely pressure?
Wow.
If they could do that, they wouldn't just have a new treatment technique. They'd have proven that, under the right conditions, physics could actually override a fundamental biological necessity. That is the real genesis of HBOT.
Okay, so the experiment itself, it's become almost legendary in medical history circles, partly because it was so conceptual pure, but also just for its sheer audacity.
Yeah.
It feels like it was designed to leave absolutely no room for doubt.
It absolutely was. The beauty of it, as you say, is in the shocking clarity of the setup. They needed a mammal, something with physiology reasonably close to humans, to show the response. So they used piglets.
Piglets, okay, and the intervention, the actual procedure. This is the part that must have made the medical establishment at the time just collectively gasp. Tell us about this extreme exchange transfusion.
Right. So the surgical team, very carefully, replaced almost all of the piglet's circulating red blood cells. They did this with what's called an exchange transfusion, basically, drawing blood out while simultaneously infusing a plasma expander solution. Think saline, plasma proteins, but crucially,
No red blood cells.
exactly, no red blood cells, and therefore, essentially, no hemoglobin.
Pause on that for a second. You take a living mammal and you remove its primary way of carrying oxygen. I mean, that induces immediate catastrophic shock. It should cause death within minutes, right? It seems like an almost impossible state to survive, even for a moment.
It really is the ultimate physiological stress test. The state they induced was acute, profound anemia so severe it was considered universally fatal. They measured the animal's hemoglobin, the HB level, down to less than 0.4 grams per deciliter. That's written as 0.4 GDL.
Less than zero point 0.4. Okay, let's do some context. A healthy human is usually somewhere around 12 to 16 GVL, maybe a bit less for women, more for men. And even in a major emergency, like massive bleeding, doctors usually start transfusing urgently if the level drops below 7 or 8 GDL. Just to survive acute blood loss, you typically need at least 4 or 5 GDL of hemoglobin still working.
Precisely. So conventional medical wisdom in 1960, and frankly, even today mostly, would say an HB level under 1.0 GDL is just flat out incompatible with life. You'd expect immediate brain hypoxia, severe metabolic acidosis, the heart just giving out because it can't possibly pump fast enough to deliver the tiny amount of oxygen left. Boerema created animals that, by all rights, should have been dead almost instantly.
So they deliberately pushed these subjects way past what everyone considered the absolute point of no return. And then came the critical step, the HBOT intervention.
Exactly. These ICA sanguinated piglets, the ones basically operating without red blood cells, were immediately put into a hyperbaric chamber. Inside, they were given 100% pure oxygen, but crucially, at 3 atmospheres absolute pressure, 3 ADA.
3 atmospheres. That pressure component is key. We're normally living at 1 ATA, sea level pressure. So this is triple that.
Triple the pressure. And this is where the mind-blowing discovery happened. The piglets survived. They lived. And not only did they survive, Boerema specifically noted they remained in excellent clinical condition. No signs of distress. Despite having virtually zero hemoglobin, they were apparently pink, healthy looking.
Wow. That is the moment of scientific proof, isn't it? It's just irrefutable. If those piglets survived without hemoglobin, oxygen must have been getting to their tissues somehow, feeding their metabolism.
So it had to be.
And since the biological carriers, the red blood cells, were essentially gone, the only other route was for the oxygen to be dissolved directly into the liquid part of the blood, the plasma itself.
Exactly. What's so stunning here isn't just the sheer nerve of the experiment, but the absolute clarity of the conclusion it forced. Boerema demonstrated that the body's huge metabolic demand for oxygen, keeping the heart beating, the brain functioning, every organ working, could be entirely met by a mechanism completely independent of hemoglobin.
The plasma, which normally just carries salts, proteins, water, it became the sole lifeline, purely because of the physics involved.
That's it. They didn't just stumble upon a potential treatment. They proved a fundamental physiological workaround for life itself. It completely redefined the known relationship between breathing, circulation, and basic survival.
Okay, so we've got this incredible, almost unbelievable experimental result. Now we have to connect it to the underlying physics, what made it possible. When those piglets survived, they weren't just lucky. They were living proof of a specific physical principle, one that scientists knew about, but maybe hadn't seen demonstrated so dramatically in a complex living system.
Right. That core principle is Henry's law. And Boerema's genius wasn't discovering the law itself. Chemists had known about it for ages. It was applying it so aggressively with such high pressure in a living mammal to achieve something revolutionary. His experiment was the definitive scientific validation of Henry's law working in vivo, in a physiological context.
So, break Henry's law down for us simply. Why does applying high pressure make oxygen do something it doesn't normally do in the blood?
Henry's law basically says that the amount of any given gas that will dissolve in a liquid is directly proportional to the partial pressure of that gas above the liquid. Simple idea, really.
Okay.
The more you squeeze that gas onto the liquid surface, the more of it gets physically pushed or forced into dissolving within the liquid itself.
Is this like the soda can analogy?
Yeah.
You seal the can under pressure, forcing carbon dioxide gas into the soda water. Then you pop the top, release the pressure.
And the gas immediately starts bubbling out, right?
Yeah.
The soda goes flat because the dissolved gas escapes. That's Henry's law in action. In the hyperbaric chamber, we're doing the same thing, essentially creating a sealed can environment for the body. But instead of CO2 and soda, it's pure oxygen and blood plasma.
Okay, so under normal conditions, here at 1 atmosphere, our plasma carries some dissolved oxygen, but not much.
Tiny amount, really insignificant in the grand scheme of things. Under normal pressure, breathing normal air, the vast majority of oxygen in our blood, something like 97% or even more, isn't dissolved. It's chemically, almost magnetically bound to the hemoglobin molecules inside our red blood cells.
Right. Hemoglobin is the dedicated oxygen bus. The plasma is just the road normally.
Pretty much. Now, this is where the numbers get really credible. This is the quantitative data that makes Boerema's proof so undeniable, the real meat of the deep dive. How much oxygen do we normally have dissolved, and how much do we actually need?
Okay, lay it on us.
Under normal conditions, 1 ATA, breathing air, the amount of oxygen physically dissolved in your plasma is only about 0.3 milliliters of O2 per deciliter of plasma. That's 0.3 mLDL, very small.
0.3. Okay. And how much oxygen does the body need, just sitting at rest to keep everything running basic metabolism?
Just to sustain life, keep your heart beating, brain thinking, basic functions going. Your tissues consume roughly 5.5 milliliters of oxygen for every deciliter of blood that flows through them. So 5.5 milliliter O2DL.
Whoa, hang on. So normally the dissolved oxygen in plasma, that 0.3 mmLA, only provides
How is that?
less than 10% of the oxygen the body actually needs, maybe around 5%.
Exactly. It's a tiny fraction. The rest has to come from the hemoglobin. That's why under normal pressure, if you lose your hemoglobin, you die very quickly. There's just no way for the plasma alone to carry enough oxygen.
Okay, so that sets the stage. Now, what happened when Boerema put those piglets at 3 ATA, 3 times normal pressure, and gave them 100% oxygen?
The combination of high pressure and pure oxygen dramatically increased the partial pressure of oxygen pushing down on the plasma. According to Henry's law, this forced a much, much higher volume of oxygen to dissolve directly into that liquid plasma.
And what was the resulting dissolved oxygen concentration at 3 ATA?
At 3 ATA, breathing 100% oxygen, the calculations and subsequent measurements show the dissolved oxygen concentration skyrockets to approximately 6.0 milliliters of oxygen per deciliter of plasma. 6.0 milliliter O2DL.
6.0. Now that's where it gets really interesting, isn't it? 6 milliliters dissolved purely because of the pressure. That's actually more than the body's minimum resting requirement of 5.5 milliliters.
That's the entire scientific justification right there in those numbers. The physical mechanism, high-pressure dissolving gas, completely bypassed and, in fact, exceeded the capacity needed from the biological carrier system, hemoglobin, for baseline survival.
Wow.
The hyperbaric oxygen environment physically forces enough oxygen into the liquid part of the blood to keep the animal alive, totally independent of whether red blood cells are there or working properly. This single quantitative reality, proven by that 1960 paper, created the entire scientific rationale for HBOT. Everything else builds on this.
And that rationale then just flows directly into thinking about clinical uses. If you've got, say, a severe crush injury or a diabetic foot ulcer with terrible circulation or a failing skin graft, places where blood flow and therefore hemoglobin-delivered oxygen is severely compromised,
Right.
HBOT provides a way to ensure that whatever minimal blood just trickle into that area is supersaturated with dissolved oxygen, enough to keep tissues alive, fight infections, start healing. It's like an acute physiological override button.
It's the ultimate compensation mechanism. It helps in ischemia, where blood flow is the problem. It helps in severe anemia, where the oxygen carrier itself is the problem. And as we'll touch on, it even works directly against certain types of bacteria that hate oxygen. It covers a lot of bases, all stemming from Henry's Law.
Okay, so we have this absolutely foundational paper from 1960, Life Without Blood. It proved the physics, it explained the survival of the piglets. Logically, you'd think, okay, every modern paper written about HBOT must start by citing Boerema, 1960. It's the origin.
You'd think so, wouldn't you? And yet, they mostly don't. Which brings us to something we could call the citation paradox. It's actually a great illustration of how science builds on itself and how knowledge gets valued differently over time. Boerema's 1960 paper is almost certainly not the most frequently cited article in the HBOT literature today. Not even close, probably.
Why is that? That seems counterintuitive. If it's the bedrock, the foundation stone, why isn't it constantly referenced?
Because we need to make a really clear distinction between a seminal foundational discovery, the kind Boerema made, and a paper used for current clinical guidance and evidence synthesis. Boerema's paper is seminal. It represents the philosophical and physiological origin of the whole idea. It answered the fundamental question, is this physically possible in a living mammal?
Right. It opened the door conceptually.
Exactly. But once that door is open and the possibility is established, the scientific and medical community moves on to the next set of questions. Questions that are much more practical, more clinical.
They stop asking, is it possible? And start asking things like, okay, does it actually work better than standard wound care for diabetic foot ulcers? Or is it safe long-term for treating radiation injury? What's the optimal pressure and duration?
Precisely. Modern medical research, especially the kind that influences practice, is overwhelmingly focused on clinical efficacy. Does it work in humans for specific conditions? Safety, cost-effectiveness, and comparing it to other treatments. Therefore, the articles that get cited over and over again today are the ones used to write hospital protocols, justify insurance coverage, and satisfy regulatory bodies.
Okay, so give us some examples. What kind of publications do get the heavy citation traffic in the HBOT field right now?
Oh, the big hitters are almost always large-scale syntheses of data. Think systematic reviews and meta-analyses, especially those published by really credible, rigorous organizations like the Cochrane collaboration. Their Cochrane database of systematic reviews is hugely influential.
Reviews, right. They pool data from lots of different studies.
Dozens, sometimes hundreds of individual studies on a specific topic. They crunch all the numbers together to provide what's considered the most definitive, statistically robust answer currently available on whether HBOT is effective for a very specific indication, like does it improve healing rates in tissue damaged by radiation? Or does it help clear up chronic bone infections that haven't responded to anything else?
So let's say I'm a clinician or a researcher at a hospital, and I'm writing up new guidelines for using HBOT to treat, I don't know, delayed radiation injury to the jawbone after cancer treatment.
No.
I'm not going to lead my bibliography with the 1960 piglet study, am I? I'm going straight to the most recent comprehensive Cochrane review that summarizes the outcomes from every decent human trial ever done on using HBOT for that specific problem.
That's exactly right. That Cochrane review, or a similar large meta-analysis, provides the current evidence base. It tells you what the accumulated human data says. That's what dictates the standard of care, what informs clinical guidelines, what insurance companies look at for reimbursement. It tells the doctor, yes, based on data from thousands of patients, this seems to be an effective and reasonably safe intervention for this specific condition. That's the synthesis clinicians and modern researchers need and cite.
It raises a really important point, though. Does this mean that foundational work, like Boerema's, becomes less valuable somehow, just because its citation count dwindles over the decades?
Absolutely not. Its value just shifts. It moves from being a direct clinical reference tool to being an intellectual cornerstone. Boerema's paper provided the essential conceptual leap, the physiological proof that allowed the entire field to even be conceived. Without that 1960 paper demonstrating possibility, none of the subsequent clinical trials would likely have ever been designed, let alone funded and carried out.
Okay, so there's a flow of knowledge here. Boerema's discovery is like the spark. That leads to early clinical applications and experiments. Those generate data for bigger, better randomized controlled trials, the RCTs. Then, people synthesize all those RCTs into meta-analyses and systematic reviews like Cochrane, and those reviews become the most heavily cited references for current practice. It's like a hierarchy.
That's a great way to put it, a hierarchy of necessity, maybe. The seminal paper is necessary for the initial leap of faith, the fundamental aha moment. The highly referenced reviews are necessary for safe, effective, and justifiable clinical deployment today. Both are critical, but Boerema's paper is the undisputed intellectual origin story. It's the moment someone flipped the light switch in a previously dark room.
What's really striking, though, is how incredibly fast they moved from that lab proof of principle to a major clinical success. It wasn't like decades of slow, cautious adoption. The physiological principles seemed so robust, they went almost straight to saving human lives with it.
That really is the hallmark of a truly profound discovery, isn't it? When the implications are so clear and the potential benefits so large. The clinical follow-up was almost immediate, and it involved one of Boerema's colleagues you mentioned earlier, Dr. W.H. Brummelkamp. He instantly grasped that Boerema's physiological proof offered a direct weapon against one of the most feared acute infections of that era.
Right, so there's another key paper, published just 1 year later, in 1961. This one documents that rapid transition from the lab bench, the piglets, straight to the patient's bedside.
That's the one. Brummelkamp, Hogendijk, and Boerema, 1961. And the title tells the story. Treatment of anaerobic infections, Clostridial myositis, by drenching the tissues with oxygen under high atmospheric pressure.
Okay, Clostridial myositis, that's the formal medical term for gas gangrene, right? For listeners who might not be familiar, what was it about gas gangrene that made it such a deadly condition and such an urgent target for this brand new, unproven therapy?
Gas gangrene is caused by bacteria from the Clostridium genus. And the defining characteristic of these particular bacteria is that they are strict anaerobes.
Meaning they hate oxygen.
They don't just hate it, they thrive, and often can only survive and multiply in environments with extremely low oxygen levels deep in damaged muscle tissue, for instance, where blood supply might be compromised. Once they get a foothold, they release potent toxins that literally dissolve muscle and tissue, producing gas bubbles as a byproduct, hence gas gangrene.
And it's notoriously fast-moving.
Terrifyingly fast. It could spread through a limb in a matter of hours. Before effective treatment, the main option was often immediate radical surgical debridement, basically cutting away all the infected tissue, which frequently meant amputation, often high up the limb, just to save the patient's life. And even then, mortality was high.
So it's this incredibly aggressive infection, fundamentally defined by its need for a low oxygen environment, racing against the clock.
Exactly. And you can see why the connection to Boerema's work was so immediate and profound for Brummelkamp. If you can use high pressure to dissolve enough oxygen into blood plasma to keep an animal alive without hemoglobin,
then you could certainly use that same pressure to force enough oxygen into damaged, poorly perfused tissue to make the environment toxic to oxygen-hating bacteria.
Precisely. Brummelkamp demonstrated that by putting patients with gas gangrene into the hyperbaric chamber, they could flood those infected tissues with such high concentrations of dissolved oxygen that it effectively stopped the Clostridium bacteria in their tracks. It arrested the spread of the infection, often non-surgically. The high oxygen tension being driven deep into the tissue by the pressure essentially turned the local environment poisonous for the anaerobic bugs.
So it wasn't just supporting the body. It was actively attacking the infection at its weak point.
Yes, it was a spectacular, often life-saving clinical triumph. It proved that HBOT wasn't just some physiological curiosity demonstrated in anemic piglets. It was a powerful, practical medical intervention. So while Boerema's 1960 paper laid the absolute physiological foundation,
Brummelkamp's 1961 paper provided the first major, undeniable life-saving clinical indication. It established gas gangrene treatment as one of the first, and still one of the few, truly emergent, universally accepted uses for HBOT that continues right up to today.
it really did.
That incredibly rapid move, proving life is possible without blood in 1960, and then successfully treating this deadly, often amputation-requiring infection in 1961, it just shows how utterly confident they must have been in the underlying physics and physiology. They didn't seem to need years of tentative small-scale trials. The principle was clear, the application seemed obvious, and the results were dramatic. It really cemented HBOT's potential in the medical world.
And what's also interesting is that while Henry's law and dissolved oxygen explain the basic survival in Boerema's piglets, the effect against the gas gangrene bacteria is arguably more complex. Yes, oxygen poisons them directly, but the hyperoxygenation also creates reactive oxygen species, free radicals within the tissue that are toxic to the bacteria and might even help deactivate their toxins. So it's like using a physical mechanism pressure to deliver chemical warfare against the microbes.
Huh, that's fascinating. So the same core principle, pressure forcing gas into liquid, is leveraged for two quite different therapeutic effects. Basic physiological oxygen delivery and compensation in the pivotal model, and then direct antimicrobial action in the gas gangrene patients.
Exactly. It highlights the versatility that comes from manipulating that fundamental physical parameter. Hashtag tag outro. So there we have it. We've successfully traced the journey of hyperbaric oxygen therapy right back to its true scientific source. We move from a known physical theory through a revolutionary physiological proof and then almost immediately into its first decisive clinical victory.
Let's just quickly recap that path we followed for you, our listener. We started with the physics. Henry's law, the rule stating pressure dictates how much gas dissolves in a liquid. Then, the crucial moment, the physiological proof. Dr. Boerema's landmark 1960 piglet study conducted at 3 atmospheres absolute pressure. That study quantified that life is possible even without functional hemoglobin, because the plasma itself under that pressure can carry enough dissolved oxygen, about 6.0 milliliter O2 to meet and even exceed the body's minimum metabolic need, roughly 5.5 milliliter O2
And that stunning physiological bedrock finding quickly, almost instantly, led to the first major clinical triumph documented in 1961. Brummelkamp's successful use of that same high-pressure oxygenation to treat deadly gas gangrene, an infection caused by bacteria that specifically require low oxygen conditions to survive.
So the key takeaway for you, diving deep with us today, is that to really understand the foundations of a complex medical field like HBOT, you often have to look beyond the currently most cited papers, the systematic reviews that guide daily practice. You need to find that original moment when the conceptual door was first kicked open. The most truly foundational discovery isn't always the one dominating today's citation counts. It's the one that fundamentally changed the scientific understanding of what's possible inside the body.
And that leads us to a final kind of provocative thought, really building on the sheer audacity of Boerema's original finding. His discovery showed that a purely physical principle, simple gas pressure, could not just assist, but could entirely override what was considered a core, non-negotiable biological necessity, the need for hemoglobin to carry oxygen. It proved that, in certain extreme situations, physics could literally dictate terms to biology.
It really makes you wonder about the limits, doesn't it?
Exactly. What other physiological systems, what other biological rules that we currently accept as absolute necessities might be possible to bypass or at least dramatically alter simply by cleverly manipulating fundamental laws of physics? Things like pressure, maybe temperature, acceleration, density, electromagnetism. Boerema didn't just invent a treatment, he fundamentally challenged the definition of mammalian survival and resilience. It's definitely something for you to mull over or perhaps explore on your own.
That is definitely food for thought. And that's all for this deep dive. Thank you for joining us as we uncover the scientific source code, the very bedrock of high-pressure medicine. We'll catch you next time.